Depression is a major health issue. It is estimated to be present at up to 17% of the US population, at least once in their lifetime, are being treated for depression. We got interested in depression when we found that a mouse which we had generated to study GABA or gamma aminobutyric acid. And we made this fortuitous observation that mice with mutations in these receptors showed virtually all the signs one would expect of a mouse that's depressed. So one of the core symptoms of depression is reduced pleasure-seeking behavior. And you can kind of model that in a rodent. They like to drink sucrose or some sweetened solution. You would expect of a mouse that's depressed that it drinks less sucrose. This is exactly what we see in our mice, and we can reverse this with antidepressant drugs. So we have this mouse model that's based on a defect in the function of GABA due to a mutation in the receptors for GABA. GABA is one of those chemicals that are produced by neurons and that serve as messengers for communication between cells. So GABA is the second most abundant type of these chemicals. The most abundant one is glutamate. And GABA and glutamate kind of work together. GABA may be considered the brake or what slows down neural activity or nerve cell activity, whereas glutamate is more like the engine or the accelerator of neural activity. And GABA and glutamate need to be in proper balance for the brain to function properly. Most biological systems, including the brain, have mechanisms to cope for such imbalances. If something is out of balance, then something else adapting or adjusting the system so it becomes into balance again. In the brain, this is GABA and glutamate. They must be in balance. If the brake doesn't work very well, the engine will be downregulated. In the case of our mice, this is achieved in part, at least, by downregulation of glutamate receptor. What we have shown is that the glutamate defect is due to a defect in GABAergic transmission. What we also have shown is that we can reverse both these defects by giving a drug known as ketamine that acts on a class of glutamate receptors. This drug does not only normalize the function of glutamate receptors, but it actually also increases the function of GABA. Thereby, we have essentially brought together two major hypotheses in the field. One hypothesis says it's glutamate that's defective in depression. Another hypothesis says it's GABA that's defective in depression. We have brought these two hypotheses together. We also have shown that ketamine does not only restore the function of glutamatergic transmission, it also restores the function of GABAergic transmission. Ketamine is a drug that has recently become of enormous interest because low concentrations of this drug have rapid antidepressant effects, and this single treatment can last for at least a week in patients. So therefore, there is enormous interest in how ketamine does that. But it's not very useful as an antidepressant drug because it's highly addictive. We'd like to understand the mechanism and use this information then to develop ketamine-like drugs that have fewer of those unwanted side effects.